like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Welcome to the National University of Singapore Yong Lin School of Medicine Healthy Longevity webinar. Good to see you, and thank you for joining. Today we will learn about mechanobiology and its relation to aging. Professor Rong Li is with us, and I'm sure her presentation will trigger lots of questions. So please use the Q&A functions to send your questions, but also, of course, your comments or other suggestions. As always, we are starting with a very short presentation today being given by Jessica Liu. Jessica is a very talented PhD student in my group at the Center for Healthy Longevity, and she will talk about longitudinal phenotypic aging metrics in the Baltimore longitudinal study of aging, which is, by the way, a great cohort. Jessica.
Thank you for the introduction. Good evening, everybody. My name is Jessica, and the paper I am presenting tonight is Longitudinal Phenotypic Aging Metrics in the Baltimore Longitudinal Study of Aging from the journal Nature Aging, published in July this year. The Baltimore Longitudinal Study of Aging, or the BLSA for short, is an observational study. In other words, no interventions are introduced. It is a clinical research program on human aging that began in 1958, and volunteers of different ages join the study when they are healthy and have follow-up visits over their life course. Hua et al. Are using data from this cohort study. This is the framework that underpins the study design of this paper. The authors hypothesized that biological mechanisms in the center square drive changes in aging phenotypes, which eventually lead to deterioration at functional levels on the outer rim. These functional levels are divided into 4 domains: physical function, cognitive function, mood and sensory, and mental health. To understand the functional outcomes, there are 4 phenotypic domains proposed for measuring the aging phenotypes, which are the colored boxes in each quadrant in a counterclockwise manner from the yellow-orange square. The domains are body composition, energetics, homeostatic mechanisms, and neuroplasticity and neurodegeneration. With these 4 domains of aging phenotypes in mind, Kuo et al. Used data collected longitudinally across the adult age range from 20s to 90s from 968 participants. The median follow-up was around 7 to 9 years. Their first objective was to develop a global longitudinal phenotypic score using biological, phenotypic, and functional metrics of aging. Their second objective was to evaluate how this score is associated with changes in health and functional outcomes. Kuo et al. Generated a global longitudinal phenotypic score based on summarizing the difference between each individual longitudinal trajectories and the population average, age and sex-specific trajectories across these 35 aging phenotypes from these 4 predefined domains. Then the authors standardized these values and averaged them within each domain. Finally, the standardized scores were averaged into a global longitudinal phenotypic score that represents the rate of phenotypic aging compared with that of the general population. They then looked at the association between this score and different functional outcomes. Here is the relationship between for 3 mobility tests. Higher scores indicate faster longitudinal age phenotypic changes, that is accelerated aging, while lower scores represent slower longitudinal age phenotypic changes compared with the overall population at the same age of the participant. While lower phenotypic scores, that is decelerated decline in aging phenotypes, were associated with slower physical function decline across all measures. Here, the authors analyzed the relationship between the score and changes in cognitive function. A higher global longitudinal phenotypic score, that is accelerated decline in aging phenotypes, was associated with faster cognitive decline. The participants experienced heterogeneous rates of phenotypic aging across a wide age range from mid-20s to 90s across the 4 phenotypic domains, which can be seen from the scattered pattern of these Black data points here and on the previous slide. When we look at multimorbidity and survival, a lower global longitudinal phenotypic score, that is decelerated decline in aging phenotypes, was associated with slower increase in the multimorbidity index on your left. Adjusting for age, sex, and education, among those surviving to at least 60 years, 1 point higher in global longitudinal phenotypic score was associated with 12% shorter survival time. In summary, accelerated longitudinal phenotypic aging is associated with faster physical and cognitive decline, faster accumulation of multimorbidity, and shorter survival. These associations studied across time are more robust compared with the use of phenotypic and epigenetic measurements at a single time point. Substantial intra-individual heterogeneity in the rate of aging exists across the phenotypic domains. And finally, the main takeaway is that combining information on the rate of aging across different domains is more powerful than relying on measures in a single domain for quantification of the overall rate of phenotypic aging. That concludes my presentation. Thank you for listening.
Thank you so much, Jessica. So I think we all should work on our phenotypic score. So I think, walk my dog after this webinar. And hopefully you're also doing some physical activity that helps for all organ systems. But before doing so, I am going to introduce Professor Rong Li, and she is affiliated with the Johns Hopkins University, where she served as the director of the Center for Cell Dynamics in the Johns Hopkins School of Medicine. And she, nearly 3 decades, was studying or is studying the cellular dynamics and mechanisms of cells. And we are very fortunate that Rong Li was recruited to the National University in Singapore in 2011. And since then, she serves as the director of the Mechanobiology Institute. So we are very fortunate to have her here and learn how we could relate that mechanobiology to the aging process. Rong Li, the floor is yours. Thank you for joining us today.
Well, thank you, Professor Mayer, and good evening, good afternoon, good morning, I guess, also. Welcome to this presentation, and it's an honor for me to tell you about our research. So let me just share the screen right now. So as Professor Mayer just introduced, I'm currently the head of the director of Mechanobiology Institute at the National University of Singapore. So this is our family photo taken recently. So NBI, short for Mechanobiology Institute, is one of the 6 Singapore Research Center of Excellence. And we were established about 13 years ago. So this is our second tranche of major funding provided by both NUS and Ministry of Education. And our research is focused on mechanobiology. So you might be wondering, what is mechanobiology? Well, mechanobiology is an interdisciplinary area of biology, focusing on the mechanical and physical aspects of biological systems. So hopefully I'll give you more impression of what mechanobiology is. So one of our focus is to understand the mechanics and dynamics of the cell. So the cell is very small, and many cells make up of our tissues, our organs. But the cell itself is the smallest self-replicating and self-organizing unit of life. So by studying the cell, we can understand a wealth of and deepest principles of biology. So in the research related to aging, we think of a cell as a kind of a machine. So you can imagine machines or cars that we see on the street, for example. As they carry out their functions, they can accumulate damage due to wear and tear, due to accidents, due to stress. But our cells actually, that also happens to our cell. But our cells actually have an enormous ability to repair and regenerate itself. So during the aging process, when the cell is young, this ability to repair and regenerate can stay on top of the wear and tear and damage accumulation. But as the cell gets old, there starts to be an imbalance. That is the repair and regenerative capacity decreases, and yet there's accumulated damage. So eventually leading to an unhealthy cell. And along this process, a very important factor is the forces that acts within the biological system, either inside the cell or outside the cell. And forces such as can create mechanical stress that leads to cell and tissue damage. At the same time, forces can also serve as signal to initiate repair and regenerative process. So what we're hoping to do is to understand how forces in biology is generated, transmitted, and also regulate diverse biological functions, especially during the aging process. So I'm actually originally I was going to tell you about two topics, but I'm going to actually, with a limited time, just focus on topic one. That is trying to understand the interplay between protein aggregation and mitochondrial dysfunction. That is as a potential root cause of age-related degenerative diseases. And this is an area of research in my own lab currently at both NBI and NUS and also Johns Hopkins. So, and before I go into the research, I also want to introduce you to a really fascinating model organism that we biologists really like to use to understand eukaryotic cells. This is a very simple creature. It's called Saccharomyces cerevisiae. It's also called baker's yeast or budding yeast. So it's the same yeast as people use to make wine or beer or bread. But it's also a fantastic model system for biologists. And here's what we can really brag about our model organism. In the past 20 years, 4 Nobel Prizes were given to scientists using yeast to understand the most fundamental processes about the cell and made some of the most important discoveries. So here we also use yeast to study aging. And the reason for that is that yeast makes aging really simple. So here's a yeast cell, and this is a mother. So the mother generates a daughter cell through this budding process. And then the mother cell can bud again and generate another daughter cell, and so on and so forth. But this mother cell cannot keep dividing forever. So after about 20 to 30 times, this mother cell starts to get old and then eventually undergo senescence and die. And yet, each time the mother cell divides, it generates a daughter cell. So even an old mother cell can generate a daughter cell that's like a newborn that has the full reproductive lifespan ahead of it. So this can be viewed as the stem cell for the entire yeast population. What this really means to us is that each time a cell divides, right along this cleavage plane, division plane, one side is the old, the other side is young. So this gives us a really simple system to try to understand what what are the determinants of aging? And how are these determinants partitioned between young and old cells? And finally, one of the questions we're really curious about is, why do cells actually need to age? Why can't they be immortal? Of course, today I won't give you the answer to that question because we don't know the answer yet. But I'll tell you a little bit about the aging determinants that we've been studying. And one of these determinants is protein aggregates. So a simplest way to explain protein aggregates is, proteins usually, as were made through transcription, translation process from our genes, they then fold into the right conformation in order for them to carry out their enzyme activities or other kinds of biological functions. But under stress conditions, a protein can unfold, and exposing areas of these proteins called the hydrophobic regions. And these regions can spontaneously interact with each other, form these protein clumps. Okay, so these are typically called protein aggregates. They're kind of protein trash in our cell, and sometimes also outside the cell. And what's really important to know about protein aggregates is that they're really well-known hallmarks of age-related diseases, such as neurodegenerative diseases, Alzheimer's disease, Parkinson's disease, so on and so forth. And also, they are also recently discovered to be present in cardiomyocytes in heart failure hearts. So the presence of protein aggregation usually reflects the cells are under stress. And the most common one is called proteotoxic stress. And so that leads to loss of protein homeostasis in the cell, or we also call it proteostasis. And other kinds of stress, such as oxidative stress or genotoxic stress or metabolic stress, can also lead to accumulation of protein aggregates. So my lab has been working on understanding a number of questions regarding to protein aggregation, how aggregation occurs and are regulated in the cell. So even young cells are able to sometimes can accumulate protein aggregates. So I just want to share with you some of the findings we published. Here are a set of results from one of my former graduate students, Chengkai Zhou, who's now an independent investigator at the Bach Institute for Aging Research. So what Kai's PhD research discovered that the protein aggregates formed in the cell, here we're using yeast as the model system, often are captured by mitochondria. So here labeled in red is mitochondria, and the green dots are protein aggregates. As you probably know, mitochondria are the cell's powerhouse. And it's also known that mitochondrial defects or damage is very commonly seen in degenerative diseases, because the cells lose their powerhouse ability to generate ATP. So here, the surprise is that mitochondria actually captures protein aggregates. And even more fascinating, what we found is that when the mother cell generates a bud, so you can see from these movies, these are live imaging of these cells. The red is mitochondria, so you can see mitochondria going into this bud cell. But surprisingly, these aggregates that were present in the mother cell are captured by mitochondria, and therefore they do not go into the bud. That allows the bud to be born, this new cell to be born with a clean slate. So based on this research, we hypothesized that the young cells have young mitochondria that carries minimum damage, whereas the old cell has older mitochondria that carries accumulated proteome damage. Another interesting facts I want to mention is that, so I mentioned even young cells can form aggregates sometimes under stress. So for example, if you give cells a heat shock, protein will form aggregates. So again, so you can see these are protein aggregates in a yeast cell. However, if we move these cells away from stress, these aggregates actually can dissolve. So the cells have the ability to get rid of these aggregates. And this is not a spontaneous process, but is mediated by a chaperone protein called HSP104, what we call a disaggregase. So if here is a mutant that's lacking this disaggregase, you can see aggregates do not go away. So this was discovered many years ago by a number of scientists, especially late Professor Susan Lindquist. Now, what we found recently was a surprise. That is that mitochondria actually has a hand in this process in dissolving the protein aggregates. So just as a way of introduction, so mitochondria is made up of lots of proteins and lipids and even nucleic acids. But the proteins in mitochondria are mostly made from nuclear encoded genes, nuclear genes. And these proteins, after translation, after made in the cytoplasm, they get imported into mitochondria to form the organelle. And this import process involves these pores called either Tom channel or the Tim channel. So what we found is that this import channel complex actually is important for protein aggregate dissolution. So that led us to propose that probably as the protein gets dissociated by the chaperones, they somehow get shredded through these pores into the mitochondria. So that's a hypothesis based on some of the observations. So we wanted to test that. And so how do we see these proteins enter mitochondria? So we use this really cool method. So you probably have all heard about the green fluorescent protein, or GFP. So GFP is this barrel-like structure made up of 11 beta strands. So usually GFP is encoded by a single polypeptide, but here it is split into two parts. The first part has the first 10 beta strand, and the second part has just the last 11th beta strand. So neither of these can be fluorescent, but only when they are come together, when they're in the same compartment, they can reconstitute a fluorescent protein. So what we did is taking advantage of this system is that we put this GFP 1 to 10, this bigger part of GFP, into mitochondria using what's called a mitochondrial targeting sequence, or MTS. So now this thing is in mitochondria sitting there, but it's not fluorescent because it's missing GFP 11. Then we tag GFP 11 to a protein that we know that aggregate. So only if this protein is imported into mitochondria, then the mitochondria will become green, and only mitochondria become green, so we can really see it. Okay, so this is a system. So we use the system to test whether aggregating proteins actually eventually gets inside mitochondria. So here's actually a piece of research data. So Fluke SM and LSG1 are two proteins that we know normally would aggregate after heat shock. 30 degrees is just a normal temperature. There's no aggregate, and you can see there's no signal in mitochondria. So the red here is mitochondria, and the green would be the fluorescent protein. And now if we give cells a heat shock, so now the protein aggregate, now you can see these proteins in mitochondria. And here's what's called super resolution imaging. So we can actually resolve the mitochondrial membranes. We can see the green signal is actually inside mitochondria. This is a 3D reconstruction. So then what happens to these proteins once they get into mitochondria? Well, the fate of that is that they get degraded. So you can see this, that after heat shock, these mitochondria accumulate green fluorescent protein. And then we move the cells away from the heat shock. So there's no more continuous import of these misfolded proteins. Then what's inside gradually disappear. They get degraded by mitochondrial proteases. So one of them that doing this job is a protein called PIM1. So in the PIM1 mutant, this process doesn't happen properly. Okay, so based on these research findings, we came up with a discovery of a pathway which my graduate students actually very playfully named this pathway MAGIC, standing for mitochondria as guardian in cytosol. So in this pathway, what we propose is that aggregated proteins, so here's a protein aggregate here, they get captured by mitochondria, and then these aggregate chaperones would disentangle these polypeptides and individually thread these polypeptides through these mitochondrial pores so that the proteins get degraded by mitochondria proteases. So this is, you probably, if you know something about protein degradation in the cell, you probably find this very strange, because we know that proteasome, for example, is a major site of protein trash disposal in the cell. It degrades most of the cellular proteins. And autophagy lysosomes are larger compartments that degrade larger intracellular unwanted structures or membrane proteins. So how does this magic fit in? So this is how we envision this third degradation system. So this is a kind of a trash disposal that Americans are probably most used to. I haven't seen this in Singapore. When I first went to US, when I saw this, I just thought it was so counterintuitive. People would be shoveling trash inside the sinkhole, but I didn't know that there's actually a hidden disposal there. So you turn on the switch, the trash gets ground out. So it's actually really neat. But one thing though, so this turns out to be very convenient. So if you have food that you want to get rid of, however, since I have a house that actually has one of these disposals, I know that it's actually very fragile. If you put too much trash in there, or you let the wrong trash go down, such as a chopstick or a piece of silverware or a hairball, this thing can be messed up really easily. So that's what we thought. Could too much trash into mitochondria actually mess up mitochondria? So this is an experiment that we did to test this. We mutated all the cytosolic chaperones, HSP70 protein. So now there's a massive misfolding, protein misfolding in the cytoplasm. And then these misfolded proteins, a lot of them, a lot more misfolded proteins now get imported into mitochondria, and they don't get cleared because there's a continuous influx of these proteins into mitochondria. Now what we see is that mitochondria actually become fragmented and also accumulate reactive oxygen species. So both of these are hallmarks of mitochondrial damage or mitochondrial dysfunction. So too much trash going into misfolded proteins going into mitochondria can really overwhelm mitochondria's normal functionality in getting rid of this trash, leading to mitochondrial dysfunction. So that leads to the relationship of these discoveries to age-related degenerative diseases, which we know have two important hallmarks. One is mitochondrial dysfunction. The other one is protein aggregation. And so it was always an interesting question of whether these two hallmarks are related to each other. So our findings suggest that perhaps they're related. Okay, so here's one of the hypotheses we had is that we know that neurodegeneration, for example, involves a protein aggregation of, for example, A beta or tau. Parkinson's disease involves aggregation of alpha-synuclein, and so on and so forth. And one of our hypotheses is that perhaps these disease-causing proteins, aggregating proteins, are actually substrates of MAGIC. That is, maybe they get into mitochondria and cause mitochondrial damage. And indeed, when we put these proteins, tag them with the split GFP system, we can clearly see them in mitochondria, as you can see in these images. And so the top panels were done in yeast. So recently, one of my postdoctoral fellows, Xi Zhang at Johns Hopkins University, actually introduced the split GFP system into mouse. So we can now see alpha-synuclein, which is the disease associated with Parkinson's, the protein associated with Parkinson's disease actually gets inside mitochondria in these dopaminergic neurons, and which are known to undergo degeneration in Parkinson's disease. And what's more interesting is that we found that the older the mice, the more dopaminergic neurons contain alpha-synuclein inside mitochondria. And she also found that alpha-synuclein can import into mitochondria can cause mitochondrial damage. So, so one of our goals then is to understand what controls this magic or import of misfolded protein into mitochondria so that we can begin to have a handle on this process. And this is, again, is a piece of work that we're just writing up. Is carried out by two very talented young graduate students, Oscar Wang and Lin Hao Ran, in my lab at Johns Hopkins. And what they did is that they used the power of yeast genetics, which provides us a read whole genome library with each of the yeast 4,000 or so non-essential genes deleted, so we can test which gene deletion actually affects this magic pathway, both leading to either leading to reduced accumulation of misfolded protein in mitochondria or enhanced accumulation. So we found about over 100 genes that regulate this pathway. And what's really surprising is that most of these genes are involved in metabolism and also involved in ribosomal biogenesis. So one of the most exciting finding is that a regulator of this process is this protein called AMPK. It's a kinase, a master regulator of cellular metabolism. So this protein in yeast is called SNF1. So when cells are grow in high glucose conditions, so there's abundance of nutrient, this kinase is not activated. But when cells are under calorie restriction or low glucose condition, SNF1 kinase gets activated, and then it's one of its function is to stimulate the expression of respiratory genes, genes that go in proteins that go into mitochondria and enhance mitochondria's oxidative phosphorylation respiratory functions. So what we found is that if SNF1 gene is deleted, so if this kinase is inactivated, then there's actually more misfolded proteins going into mitochondria. So the magic is enhanced. But if we increase the kinase activity by deleting a phosphatase that normally inactivates this kinase, so now cells have overactive high level of AMPK. So now this magic pathway is repressed. So there's very little misfolded proteins going into mitochondria. And we found that this ability of AMPK to turn off this magic pathway is actually protective of mitochondria when there's plenty of proteotoxic stress. So what we this finding suggests that AMPK is a switch that controls mitochondria's function in energy production, which we know is its main function, but also this moonlighting function in proteostasis. So when there's abundance of glucose, where allows cells to generate ATP using using glycolysis, the magic level is high. So basically, mitochondria perhaps have an enhanced activity towards helping to get rid of misfolded proteins. But when the glucose level is low or AMPK level is high, then mitochondria is the magic function is shut down, and then mitochondria perhaps has its main focus in generating ATP through oxidative phosphorylation. So finally, just to summarize what I told you today, so we've our research have generated some really useful insights, perhaps, for understanding degenerative diseases. And we know that there are two hallmarks of these diseases: mitochondrial dysfunction and protein aggregation. And we believe that really have a very close relationship. So our research suggests that mitochondrial dysfunction can help the accumulation, can lead to accumulation of protein aggregates, or preventing the clearance of protein aggregates, whereas cellular stress can promote protein aggregation. And if there's too much protein aggregation, too much gets into mitochondria, then that leads to mitochondrial dysfunction. And these two arrows, you can look at it simultaneously. If they happen simultaneously, then it can form a vicious cycle, perhaps leading to degeneration and precipitate diseases. So the goal of our research then is to understand how we can break this vicious cycle in order to delay aging and prevent or cure degenerative diseases. Thank you.
Thank you so much, Professor Rong Li. This was a magic presentation, I would say. I already think about, can we transplant mitochondria, etc. But let's start a little bit on a higher level. May I ask you, if you're looking at the protein aggregation between species, because you really focused on yeast, etc.
Right.
Is that translatable to other species and to humans?
Yeah, so I didn't have time to show you, so we have quite a bit of data now. Also, after we published our first study, other labs have also been looking at this in human cells. So there are quite a bit of information about that. And so, for example, we've seen misfolded proteins going into human cell mitochondria, and the more proteins that are more prone to misfolding, the more likely they accumulate in mitochondria. Of course, not every protein has the ability to go into mitochondria. So we're actually trying to understand what are the rules and regulations that control which misfolded proteins go into mitochondria. But entry of misfolded proteins into mitochondria has been observed. In fact, there's a wealth of literature now about some of these neurodegenerative disease proteins going into mitochondria and causing mitochondrial dysfunction. So these are probably the bad trash that go into your sink disposal, the hairballs, the chopsticks that shouldn't be there, and they go in there and they mess up things.
That's fascinating.
Yeah, so that one piece of data I showed you in mouse, that's mouse own alpha-synuclein protein. And so we just put this split GFP system so we can see it in mitochondria. Without this trick, it's hard to see these proteins in mitochondria because they are also, very highly present in the cytoplasm. So it's, you can imagine it's a sea of these molecules. So and mitochondria is very narrow, so it's very hard to see it. So using our system, we can block out all the signals outside of mitochondria, so we can really just see what's inside.
Yeah. So and is it then dependent also on which cell you are looking at in a certain species, or so is the trash mitochondria in a certain cell is different in different cell types?
Yeah. So when we look at this in mouse, basically this is using transgenic techniques. So literally every mouse cell have these introduced genes. But we don't see this in mitochondria in every cell, and we don't. And as I mentioned, the dopaminergic neurons in the midbrain region somehow accumulate alpha-synuclein in mitochondria. We don't know why. So we don't know why only some cells are affected, but other cells are not. And one possibility is that, as I mentioned, there's a wealth of proteases in mitochondria that can degrade these proteins. So many cells maybe just have a very robust ability to degrade these misfolded proteins once they enter mitochondria. But maybe some cells, for reasons we don't understand, have weak ability to manage these imported misfolded proteins.
Yeah, so it's cell type specific, and then it's also specific to the age a little bit of that cell. If it's a young cell and it's an old cell. So But do you really see in your experiments that it's either young or it's either old, or as we see clinically, that aging is more a trajectory of decline in function?
Yeah.
So do you see
it's
an
a trajectory?
Yes.
No, it's not. It's an increase, although there's a kind of a sharp increase towards the in very old mice. So
yeah,
there's definitely a gradual increase in the beginning.
And is that decline or the increase of the losing capacity to deal with the trash, is that dependent on the cell type, or is it the rate of, I would say, aging in that cell? Is that a little bit the same in each cell and each organ maybe?
Yeah, they're different, and also they seem to have different ability to deal with different trash. So for example, in heart failure cardiomyocytes, there are also protein aggregates accumulating, and those are different proteins. They're not the same proteins that cause neurodegeneration. So right now we actually have a new large collaborative project between NBI and the Cardiovascular Institute at NUS to really understand what are these proteins that aggregate in cardiomyocytes in the aging heart. One of the interesting idea is that these proteins are actually involved in fourth generation, and when they're constantly under force or abnormal force, they tend to get unfolded and perhaps undergo the aggregation process. So that's a hypothesis that we're currently testing.
Okay, so I did lots of culture experiments in my past, looking at fibroblasts, etc. And we wanted to look also at mitochondrial function.
Yeah.
But most importantly, and that was like 10, 15 years ago, people said, no, you have to look at the number of mitochondria and not so much, or at the balance between function and numbers. And we know that the numbers are changing during the aging process and differently also in different age groups. Can you tell us a little bit how there should be a balance between the number and the function of mitochondria when we are talking about removing trash?
Yeah, I mean, there's a lot that's not understood. Mitochondrial numbers depend on the pathways that control mitochondrial biogenesis, and that respond to, for example, energy state, metabolic state. This AMPK protein that I mentioned regulates mitochondrial biogenesis. It can cause mitochondrial number to increase, but it's not just a number. Also, mitochondrial quality has to be good. Has to have active enzymes that carry out different metabolic processes, and also in ATP production, in maintaining membrane potential, in importing all these useful important essential enzymes or building blocks of mitochondria. So all of that has to be working well, and then mitochondria also has its own genome. And although mitochondria are essential for the life of a cell, the genome can be lost, and mitochondria can still have its essential function, but it cannot carry out oxidative phosphorylation. So, and loss of mitochondrial genome has also been seen in degenerative diseases. So mitochondria's ability to maintain a genome also can deteriorate during the aging process. So there's a lot about mitochondria where really, the field is very interested in understanding how can we keep our mitochondria healthy. That's really important because that's our cell's powerhouse.
Yeah, it seems so. So we had lots of debate in the past. Are mitochondria really so important? And I think I'm really convinced now. But especially in the balance, finding how much is being invested in the proteostasis versus energy production. Yeah. And that AMPK link is very interesting. And of course, the audience might hear, okay, there's glucose. What does it have to do with diabetes? And is that the reason why diabetics have so many other age-related diseases which are actually associated with protein folding, misfolding, etc.?
Yeah, so it gives us a lot of ideas. Of course, we can just assume that's the case, but at least it gives these basic discoveries really give people new ideas, new angles to look at the specific diseases that they're interested in, to explore these, possible links and perhaps discover novel targets for treatments. So that's why we work on very fundamental discoveries about the cell, about tissues. But we believe that our discovery is really the foundation for the most effective ways to delay aging and treat age-related diseases.
Yeah, which brings lots of hope, of course, for age-related diseases, which are very chronic, like dementia, and you mentioned Alzheimer's disease. But before going to Sangeeta, I think, who has lots of questions from the audience too, let me just ask you and pick your brain with my last question. I'm a clinician doing lots of bench research too. So if you would dream in the next coming 10 years, magically, how could your finding translate into something I could use in clinical medicine and clinical practice? Yeah, so for example,
with relating to Parkinson's disease, we're trying to understand, how does alpha-synuclein, which is really one of the focal point of this disease, alpha-synuclein is known to be really the main disease protein for one of the main disease proteins for Parkinson's disease. If we understand what is the process that gets alpha-synuclein into the mitochondria, and then how this mitochondria normally clears this protein, and why some in disease cells, why this process is not working well, then we can for example, we can boost the activity of proteases that degrade alpha-synuclein, or we can shut down the process that import alpha-synuclein into mitochondria, and this could either slow down Parkinson's disease. I think for these degenerative diseases, really my naive view is that early detection and prevention or delayed disease progression is really the key. So I believe that these discoveries are very, very valuable for translational work and eventually introducing new treatments into the clinics.
Yeah, absolutely. I think we share the vision that prevention is much better than the current healthcare system, where it's already a little bit late, I would say. So that's great. Thank you so much, Sangeeta. Sangeeta, by the way, is a research assistant in our Center for Healthy Longevity. Very talented. Sangeeta, what do we have in terms of questions from the audience?
Thanks, Andrea. Hello, Prof Rong Li.
Yeah.
Thank you for the interesting presentation. So I have a few questions from the floor for you. So the first question is going to be, what are some practical lifestyle modifications one can do to reduce proteotoxic stress during aging?
During aging? Well, that's a great question. That's something that we scientists are hoping to get at. Right now, I'm not aware of any specific drugs or treatment that can help reducing that. But if you think about the mechanisms of how protein folding is or misfolding are controlled, so presumably enhancing chaperone activities or preventing proteotoxic stress that cause protein to misfold, and then enhancing the ability for cells to get rid of these misfolded proteins to basically dispose trash in a timely manner. Those are probably ways for intervention. Does that answer your question?
Okay.
Okay.
Okay. So now I'll move on to the second question. Yeah. So why do cells use mitochondria to handle misfold proteins but not other organelles? In other words, what is the advantage to do so?
Yeah, that's a great question. It's kind of asking why evolution does certain things this way. We can't really, it's hard to, we can guess. I can give you my fantasy answer. answer that I don't really have a proof for, but it makes sense. So the origin of mitochondria is such that they're thought to be symbiotic bacteria inside a primordial eukaryotic cell. So possibly, this is a mitochondria helping the cell getting rid of protein trash is one of those symbiotic underlies the symbiotic relationship. So on one hand, mitochondria helps the protein getting rid of help the cell getting rid of the unwanted protein. At the same time, these proteins, once they get degraded and turned into amino acids, it gives food to the bacteria, this symbiotic bacteria, and maybe the amino acid can be used for biosynthetic purposes or even energy production. So that could be a very ancient, process that's left over. That's still happening. So that's just a guess. But is there advantage to this? That's why I have this analogy of the system. It's kind of like the sink disposal, right? There are people who love it and there are people who hate it because it's finicky. But at the same time, it can be very convenient.
Okay, I will move on to the next question. So do cytoplasmic proteins enter mitochondria via aggregating to mitochondrial protein that are naturally directed to mitochondria?
So that's an interesting question. Okay, so in these protein aggregates that are captured at mitochondrial surface, they do contain, some of them are proteins that normally would go into mitochondria, and they're very aggregation prone. So they're there, and we don't know whether they act as leaders to draw these proteins, the aggregates, to the surface mitochondria or play any role in the mitochondrial import. But I really need to say is that these aggregated proteins do not go in as a clump. They have to be disaggregated by the chaperone protein so that they become unfolded polypeptides. That's how they can get threaded through these pores. So the chaperone function is very important for getting these protein inside mitochondria.
Thank you. So moving on to the next question.
Okay.
How is the magic process, import and degradation affected by the senescence?
Great question. We haven't looked at it. So really, this discovery is still pretty new. There's a ton of things to do to look at. So that's a great question. How is this, in general, how is this pathway or other proteostasis pathways affected during the aging process? How do they compare in old cells versus young cells? And I'm sure there's going to be cell type dependent alterations, or depending on the stress type of stress that a type of cell or tissue that normally encounter. So it's very interesting. I think overall the aging research, when it comes down to molecular and cellular level, there's a huge amount of mysteries and discoveries to be made.
Okay, and then another question from the ground. So what are the major causes of proteotoxic stress that can lead to dysfunctional mitochondria during aging?
Yeah, so one well-known kind of stress is oxidative stress. So protein oxidation can lead to protein misfolding and protein aggregation. Heat stress is another kind of stress. And there's, even though it's very commonly used in experimental settings to induce protein aggregation, it's not so clear actually in physiological conditions in mammals, for example, whether that actually play a role in causing proteotoxic stress. Also, another thing that can cause proteotoxic stress is if there's some deterioration of protein degradation system. So if you have problems affecting the proteasome, and that would cause accumulation of a lot of misfolded proteins in the cytoplasm because they get cleared too slowly, and then kinetically that would more favor protein aggregation and can cause the accumulation and lots of other protein degradation system, autophagy, and we have shown magic can also prevent cells from readily clear out or dissolve these aggregates.
Okay. And then we have one more question. So, do the protein aggregates have the preference for healthy mitochondria or damaged mitochondria? Could magic link or active to mito-UPR?
Yeah, so that's a really interesting question. We don't know yet. We do know that by accumulate. So we have another study that I didn't have time to show you. Is that protein aggregates can also accumulate inside mitochondria and cause mitochondrial damage. And so we definitely know that mitochondria that carries a lot of these aggregates tend to be damaged. But how does already damaged mitochondria interact with aggregates? That's something we haven't looked very deeply into.
Another one more question for you.
Okay.
What does one do to keep the mitochondria working well as we age?
I can say exercise is probably one way to keep your mitochondria healthy. And that's one thing I really, believe. Another thing is, I'm just speculating here. For example, our research suggests that, under low glucose condition, mitochondria really limit the amount of misfolded proteins from going in, and that has a protective effect. So maybe reduce sugar intake and keep your mitochondria busy doing oxidative phosphorylation might be a good way to keep mitochondria healthy.
Okay.
This is just a speculation.
I think this is a very nice speculation, ending with lifestyle approaches, how we can age more gracefully and maybe hopefully also healthier. Right. This was fascinating. Thank you so much for your insights of trash and how magic could actually help. Please, the audience, please send us still your questions and we will forward them. But we would also, of course, really like to hear your thoughts about which speakers we should invite for the next sessions. So use the panelists and all attendees option to give us feedback and leave your comments. So once again, Professor Rong Li, so it was a great talk. Thank you so much. For the audience, we of course have always news, and we are very active at the moment on Twitter and LinkedIn. We also have a new website, so please feel free to visit our website and follow us on the social media. We also have news which we will give at the end credits after the final video we are going to present. You might want to know what we are going to do next week. I will present also next week together with Dr. David Fehrenbach, and he is a great researcher from the University from Edinburgh. And instead of focusing on mitochondria, we will focus on kidney and how senescence might interfere with kidney diseases at older age and the decline in kidney function. As always, I will leave you with a final video, and it's about, it's about CrossFit. And I would say the technique matters. Take care.
springtime. Like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.